Namaskar, Swagatam. We would like to welcome you all on our 522nd journey of brainstorming, knowledge sharing, and inspiring for next generation. Thank you very much for joining as a speaker, as an audience, as a resource person. Thank you very much. Now, I would like to request Professor V.K. Agnihotri sir for his initial blessings. Professor V.K. Agnihotri.
Thank you, Professor Pawan Sharma ji. Namaskar. It's our great pleasure to organize 522nd webinar in the continuation, and today's topic is very charming, that is System of Clinical Research for AYUSH. So on this occasion, we welcome all our distinguished guest speakers right from very beginning. The first guest speaker of today's webinar is Dr. Renu Rathi. She is Professor and Head, MG College, Wardha, in Pediatric Department, in Kumar Bharti Department. Welcome, ma'am. Our second speaker of today's webinar is Dr. Rajeshwari PN. She is also Professor Department of Shalya Tantra in Amrita College of Ayurveda, that is from Kollam. And again, our third speaker is Dr. Payodhi Dharji, and he is from Calcutta, along with Dr. Arun Raj GR, Department of Kumar, again SDM College, Hassan. So we have got a very great platform. And when the matter of clinical assessment and clinical research is concerned, since the life starts right from the infants, so we are fortunate enough that our two pediatricians are with us. They will throw light over the day one or the hour one or the minute one of the life, which is starting with the infants to the leading to the surgery too. If at all any ailment is happened and the conservative treatment by Kaya Chikitsa department could not succeed in that way, then comes the role of our surgeons. So we are fortunate enough and we are rich enough to have the galaxy of right from Balrog to Shalya. So we do hope that today's webinar is going to be very good and full of knowledge. We all, those who have joined along with our distinguished guest speakers, with our Dr. C.P. Sharma sir, Dr. Hari Sharma ji, Dr. Mahesh Sharma ji, along with our Mamta Sharma ji, Poonam Gupta ji, Sumod Karji, and those again who are just going to join by the start of our webinar or till end. We all welcome them, along with those who will see the live program on YouTube, and again those who will visit our archives also. So thank you very much, and we welcome from our side. Thank you. Over to Pawan Sharma.
Thank you very much, sir. We would like to invite Dr. Renu Rathi ji. First of all, welcome Dr. Renu ji. As cold is there, winter is there, Dr. Agnihotri ji every time is reminding me that temperature is 7 or 8 degrees centigrade in Haridwar. So I am humbly requesting to all speakers that time limit is 20 minutes. So please kindly consider for that. Thank you very much.
I am covering by 5 layers. 5 layers.
Excellent.
Along with, I've just removed the cap and this is very much with me just to keep ourselves protected. It's 7 and fog is there. Early morning fog.
And now we all should take care, sir. BF7 is coming.
Yes.
Welcome, Renu ji. Welcome. Platform is yours. Kindly unmute yourself. Yes.
Good evening, all.
Good evening.
Yeah, topic is different designs of clinical research. So I'll just make a presentation mode. Yeah. We will have a short relook on definition of research, then its types, then categorization of clinical trials, because we are dealing with clinical research. So we will, in short, we will look into bias and errors in design. Then what is RCT, that is the topmost gold standard research design, that is called as RCT. And then other designs we will touch upon it, because time is too short to discuss on these all study different designs. So we will just touch and we'll go fast. As we all know that research is especially and search, that is scientific and systematic search for pertinent information on a specific topic from known to unknown. So the types, if we discuss about the types, then we have to see first of all that descriptive research, in which surveys and fact-finding inquiries to be incorporated, and then analytical research, in which 2 to 3 in facts or informations are already available, and we need to analyze them to make a conclusion that which one is good or more good. Then applied or fundamental research, in which we have to just focus on concept or any fundamental principles to go ahead with the with its specific motive to work upon it. And then fundamental research is specifically theory to work upon it and to go towards the concept, final concept. Then we can divide the research into is a quantitative and qualitative also. Quantitative, in which we are we can just find out the one particular figure, and in qualitative there is a gradation like mild, moderate, severe, or a gradation as severe to like Likert scale, or like that. Then clinical research, which is very much important, or diagnostic research, in which vaccines or any diagnostic tool, or we need to find out that which one is a good one, or as per the new diseases are coming, so we need to keep an eye over diagnostic research also. Then exploratory research for to make a hypothesis, to just develop a hypothesis or make a hypothesis, rather than its testing, because testing is related with the experimental trial, which is related to clinical research. And then specifically there must be a why, must be there, that intense quest of learning, or should be there, that why we are dealing with this research. There must be some interest, then only we can go ahead. So there are certain tools that we have to find out our interest, as well as scientific merit, or self interest, or commercial value, or future gain must be there, or then practical consideration. And we need to after a decision on one particular topic, then we have to go through good literary research and other different data or indexes, that is PubMed, Metacrawler, or Dogpile, or Profusion, or ZepMeta, etc. And we need to search again with the help of review articles and textbook too. When we just do the confirm on the one particular topic, then we need to go through the review of literature, and then we need to formulate the hypothesis. Before the formulation of hypothesis, we need to search gap analysis. Whatever researches are already being done, then we need to find out what else we are making gap fulfillment, or making it more beautifully designed, or inference should become, or outcome should be there. Then only we can go ahead with that gap analysis, and then formulate our hypothesis. And then we need to go ahead with collection of data, analyze of our data, and interpretation, and then final conclusion must be there. Then the whole clinical research must be based on ethics, that we must know that there must be some stages that which those which we must cross, like IEC, then CTRI, then whatever there is a bias or any kind of confounding factors, we need to eliminate them. Then only the clinical research will be more good. So we have to stick on so many guidelines, like first of all GCP ICH guidelines, then ICMR has been from 1996 we have given ICH GCP guideline, then ICMR have time to time given so many guidelines. Then as per in equator.net org, there are so many variety of guidelines, like for RCT there is a consort guideline, for review articles or meta-analysis or systematic review we have Prisma, then for case report we have care guidelines, then we have so many other like observational studies, stroke guidelines. So we have to stick on that those guidelines, or checklist is there, and as per checklist we have to go ahead with that research. Then next is there are different traditional designs also, like crossover design, factorial design, add-on design. But we are now, I'm not dealing with all those because time constraint is there. So just we will go ahead with the RCT only. That as per the revised Schedule Y of the Drugs Cosmetic Act 2005, we need to specifically treat with clinical trials with ethics committee and eco format. Especially, we need to plan it very systematically, because it is a proper proverb that failing to plan is planning to fail. So we need to have patience, positivity, as well as perseverance. Then only we can go ahead with overcoming of all other hurdles. There must be control group as a comparison to go whichever is the standard guideline already established. Then we need to compare the trial drug with control group, and we have to find out which one is better. So there is a categorization from one center study to multi-center studies, and if multi-center is there, then all we have to divide the multi-center studies into different zones, like south, west, east, and west, or central. Like that, 4-5 zones we can divide, and the multi-center to represent the data from that particular population. And also we can divide it into national and international. So if we are coming to bias, because bias is important tool in clinical research, in which we have to eliminate them by certain guidelines, by certain rules and regulations to minimize it. Then for that, specially randomization is there. Then different, I'll not go through it in detail, because there is a, in short, I just touching it on that is the information bias, or recruitment bias, or confounding bias, or outcome bias. Because if we know, or we are just biasing the things that if we know that this particular person is my known, then I'm adding to trial group only and not in control group, then it is a bias. So there must be randomization to minimize the bias. Then also blinding is a very good tool in which we can reduce the bias, and monitoring the clinical trial in thorough ways by suppose whosoever in funding agency, like AYUSH is funding agency, or whosoever is funding agency, that inspection team must monitor it every time or time to time to go through it, that whether the clinical trial is going properly or not, or there are certain flaws, or we need to eliminate them time to time. Then there must be thorough data management, and data verification must be there as per quality control and quality assurance procedures. Then there are different types of randomization, like simple, block, stratified, or cluster randomization. Then if we see in figure in this pictorial cube, that there are in these two black shadows are there and only one white. But in this like picture, whenever we randomize them, then it must be very equal amount of quantity, equal, that everyone must get chance to come into the study design. So whatever population is there, we need to pick up those particular size or participants to make them equal, equally male or female, or equal race, or equal gender, or equal religion, or like that, or we can make it as a stratified also. Then fundamentals of randomization is particularly it is unable to predict the group that whichever patient or as a participant is known to us, that must not be ambiguously registered, or it should be as per computerized generated table, or particularly stick on to the type of randomization. Then researcher must be unable to change the patient's allocation, because there must be allocation concealment in which blinding is there, and blinding is a single blind, double blind, or triple blind. Then we must be eliminating the bias, and the study clinical research must be in a very good quality. Then techniques, we will go fast. Then the benefits of randomization, particularly there is a independency, and variables are likely to be arranged in a distribution manner, and statistical analysis is in good position by biostatistician, and thereby there is a no bias due to randomization, and the clinical research will be in standard position. Then blinding, we have discussed that there is a single blind, double blind, and triple blind, and maybe quadruple blind. But in particularly Ayurveda, we can say that sometimes there is a difficult to make them blinding, because herbal formulations are not enough matching with each other, that we can blind them, because blinding considered a particular condition that the two packs must be very similar to each other, and there must not be any, means we have to put code, and we have to make them in envelope quoted, that we, means either researcher or participants could not make it guess that which one is which intervention is going on. So there is a, in short, we have discussed blinding, which is a having good assessment, protects from assessment bias, and checks placebo effects too. But there are certain limitations, that logistically complexity is there, ethical complexity is there. That's why sometimes in Ayurveda there is a blinding is not possible also. In clinical research, we have to stick on guidelines particularly, and there must be a superiority trial or inferiority trial or equivalence trial. According to this gradation of trials only, we have to choose the particular sample size, and we have to go ahead with assumption of that particular amount of percentage of efficacy we are expecting, and accordingly that we have to go ahead. And in clinical trial, I must say that many times there is a myth that size is considered on population, but in clinical trial particularly, there the size calculation must be on effect size. That is, whatever research is already completed or already published, from that particular published data, we have to took out the standard deviation and size, and as per based on standard deviation, we have to calculate the effect size. And then we have to go ahead as per the division of superiority. If we are expecting more result, then it is a superiority. Then if we are expecting equivalent result, then it is an equivalent trial. And if we are not expecting that much good efficacy, then it is an inferiority trial. But generally we do only superiority trial or equivalence trial. So we have to ask certain questions, that whether it is a exposure is there or not. If yes, then it is an experimental study. If exposure is no, then it is an observational study. It is very so as simple as that. But whenever we say that intervention is S or exposure is S, then we need to find out again randomization, allocation, concealment, and accordingly after randomization, we have to go ahead. And if it is a non-randomized, then it is called as CASI trial. And if it is an observational study, we have to stick on that. That is a cohort study, means a group of participants, like one village or one whole society or one whole big area. We can call it as a cohort. And if we have to check their prospective results or retrospective results or mixed also, we have to guess that whether this particular like intervention is more capable to treat the result, or whatever already disease has been already been as occurred, then we have to go retrospectively that which drug has been worked out for to treat this condition. As in cohort, both the policies are okay. Then in analytical study, we have to again divide it into case control as well as cross-sectional, and another way is descriptive. In classification of clinical trials, we can divide it into a treatment trial as per the motive, as per the purpose of the trial. We can divide it into a treatment trials, prevention trials, screening trials, or quality of life improvement trials, or compassionate use trials, or genetics trial. Whatever may be the purpose, we can rename it. Classification as per design: parallel group trials, crossover trials, factorial design, randomized withdrawal design, adaptive design, or sequential design. So particularly RCT is more important. So we have to just one more focus about RCT only, that it must have randomization to eliminate the bias, and there must be standard therapy, standard control, as compared to just blank control or any kind of placebo. Instead of that, we have to choose standard control. Thereby we can compare it with the trial, and size calculation must be there as per effect size. Then consent must be there, or assent if children is there. So children have not considered as a well effectively decision maker. So we have to confirm the consent as per the caretaker or parents. And then IC must be there, that is institutional ethics committee must be there, that it should be go thoroughly to eliminate any kind of bias or non-harm thing, or any kind of justice or integrity or beneficial should be taken into consideration. And then as per these tools, we have to fix our study design as RCT. Then we have to eliminate the especially run out period, or whatever previous intervention is there. We have to just make a gap of certain days, 15 days or 1 month, and then we have to restart the next intervention. Otherwise it will be a crossover or efficacy. That's why we have to keep certain gap between two interventions. And we have to treat a certain duration carryover effects, that is a carryover effect. We have discussed run-in period. Placebo controlled trial are more actually nowadays. Plus, instead of placebo, there must be standard standard therapy to check it out as a previously standard therapy is more good, or newly invented innovative trial is more good. So we need to compare it. Then crossover design is particularly one certain intervention is there, then we need to give certain gap, and after that gap again we have to again we have to give that particular intervention after certain gap. Means we have to alternatively if two groups are there, we have to just make a crossover, that so after certain gaps we have to replace that drug. It is very clearly depicted in this picture that drug A without drug A is given in group A, then washout period means certain gap must be there, and after drug B has to be given, and then in group B particularly drug B is first and then drug A is first. Means both intervention should be there in both the groups, but there must be certain washout period. So another is factorial design, in which one group is given one particular intervention, another group is given another intervention, and then in another third group we can combine both A and B interventions, and in another group we can just make it as a blank control to check out the efficacy. So these are the factors in which we can divide it as a factorial design, in which we have to check the efficacy of that particular intervention, whether it is more good, or combination is more good, or no intervention is more good, or particular A or B is more good. So particularly why research is essential in Ayurveda, because Ayurveda is naturally going globalization, and globe is taking patent one by one, and we are just waiting for them. And then like haldi and like so many things, we are just this is not good, because we need to make our Ayurveda more strong and globalized way. That's why we need research. Our updation must be there in Ayurveda, and that's why clinical research is more authentic. Then only we can, because modern science is waiting for certain evidences. Evidence-based Ayurveda. That's why we have to prove with certain facts and evidences. Then only we can firmly firmly in position to say that this is the certain facts, and that's why Ayurveda is more good. So we have that evidences in our hand. That's why research is must in Ayurveda. Then only we can publicize our Ayurveda in a safe, cheap, and quickly effective interventions, because there are so many myths regarding Ayurveda. We need to eliminate them by doing clinical research. So there are 5 phases. We will shortly discuss phase 0. That is whenever there is a new, because Ayurveda is not new, or neither it is a new molecule or like that. So we can directly, it's the Aptopadesh in our granthas. That's why we have to take one particular formulation, and we have to just as a reverse pharmacology, or like we have to just again for certain facts and evidences, we need to research it, and again establish the facts with the phase 0, like less sample size. Then phase 1, 15 to 30 participants only. Then phase 3, phase 2, certain few hundreds, and after phase 3, for few hundred, more than few hundred, and then phase 4, like certain 800 to more than thousands. In this way, we have to increase the sample size or participants, and we have to establish that this is more good. So in that way, summary, the ultimate goal of clinician is to help patients live long and functional and pain-free. So by adopting the principles of evidence-based medicine, it is maximized to patient care and stick to GCP guidelines, as well as we have to stick on equator.net org guidelines, like Consort or Prisma, or all care guidelines, and we have to establish our formulations or our Ayurveda in a new, innovative, and evidence-based way. So there must be, we can publish our data in Scopus or PubMed, or like highly indexed journals like Q1 to Q4, or in this way we have to just put up our data in a very authentic way. Then only modern side think that yes, Ayurveda has certain more good things, that then only it can be considerable. So naturally, clinical research must have robust trial design, and we need to have patience and perseverance. Then only we can go ahead. Thank you all for giving me the opportunity and patient listening. If any kind of any queries, I would love to answer them. Thank you.
Thank you. Thank you, Renuji. Thank you very much. You have covered almost all the things. Excellent. Excellent in a broad way. Great. Agnihotri, sir.
Namaskar, Pawanji. Thank you, Dr. Renu Rathiji. You very intelligently narrated all the sectors where the research work is going on, and what are the protocols, what are the rules and regulations which are set to be observed. Then only it will be said that it is a final work and given the importance. I was in dilemma that, well, I am again attached with the research work right from 1980-81 until date, and all the colleges, all the great institutes are taking out this opportunity to go for research. But simply, I was in dilemma that will there be any possibility for the research in it? I was actually in mood to listen something that the topic is system of clinical research for AYUSH. So for AYUSH purpose, will there be any parameters of Ayurveda to be researched? Because when the literature was written, it was not like that. It is coming from Satyug to everything, and of course from Vedas. So I may expect something that will there be any, or is there any possibility to go for any Sanjini Vati or anything, or Sivankirti Ras or Sitopladi Chun to be researched with the available say literature, and then be given a documentary proof that yes, it is evidented. So persons of other walk of life may accept it, or rather should accept it. Anyway, thank you so much for your very intelligent work and mission.
Sir, actually, so many pramanas are there, like Aptopadesh, Pratyaksha, Anuman, Yukti. So many pramanas are there, but they are they are unknown to all globe. So that's why we have less consideration, and that's why we need to.
Okay. Let us have the opinion of other persons, those who are coming to our platform today as the distinguished guest speakers. Because if we Ayurvedic persons will not talk on these lines, who will talk?
Yes,
how long we will rush after them for just for the evidence? So thank you so much for your intelligent presentation, which covered all.
Thank you very much, sir.
Yes.
So now I would like to
excuse me,
invite.
I just have a question.
Question and answer in the last, sir.
Okay.
That will be in the last, sir.
All right. Thank you so much.
After all speakers, we will conduct question and answer, sir. So I would like to invite Dr. Rajeshwari PN. Welcome, Dr. Rajeshwari from Amrita School of Ayurveda, Kollam. Welcome.
Thank you, sir. Am I audible? Good evening, everyone.
Yes, good evening.
Yes. Okay. Very good evening. So I have come up with a very small topic, that is system of clinical research for AYUSH. Because as a clinician, how I can project clinical research was my challenge. So what are the challenges I faced and how to overcome those, and what are the recent updates are available. So those things I'm just summing it up in a very brief slides. And organizers, kindly excuse, I'm not well as such because my voice is not coming up. So I'll yeah, okay. Thank you. So recently, when we were just going through a lot of papers for the clinical trials and to design them, and also what are the challenges we are having. This was a one clinical trial in Ayurveda, issues, challenges, and approaches, and editorial writing, which was found, and it took us to a very interesting area where we are lacking in research like that, and what are the challenges really we are having as Ayurveda doctors, and which are the subjects that has to be taken into more consideration towards the research, etc., were discussed in this paper. So based on that, in the conclusion also they are telling, is there any standard tool or standard method for researching Ayurveda medicines? So when that problem was asked, some of the literatures which gives us for our knowledge is whether these are the controlled designs or controlled trials which can be used for traditional methods. It's a black box design, stepped wedge design, and in one of the one design. So these are, I would say that it is a very much favorite topics or favorite designs which an Ayurveda doctor can adopt, because it's very simple and very easier, and we all can start our research from this level. So the black box design, it says that whether Ayurveda medicine is effective in a particular disease. Okay, So we are all running behind this, and we all need this rather than many of us. We know we are all in academics, and we'll be as also we'll be a clinicians. Most of us may not be able to put effort on those areas where we need a randomized controlled trials, etc. So those people can, or individually we can take up this kind of studies. It's a simple pre and post test like designs you can take whether Ayurveda medicine is effective or not. So another designs which they suggest us is a stepped wedge design. It's efficacy of an intervention what we are administering over a time. Whether if I am giving a Triphala Gugulu for about 1 week, what is the benefit? If I am giving it for 2 weeks, what is the benefit? If I am giving it for 4 weeks, what is the benefit? So if I am looking into all these aspects, then also it's going to give us a good amount of information. So those points are assessed by a stepped wedge designs. And this is how it comes. A medicine is given, and how in a 1 day, in 1 week, 2 weeks, 3 weeks, 4 weeks, and all in a successive weeks, what will be its effects. So the other one is of N of 1 design, means that the sample size is only 1. So this is very easy for all the clinicians. So I'll give you an example how we can adopt for an Ayurveda and how we can use our Ayurveda tools here. See, in here, we are not taking into any groups. We are not going for any of the divisions, group division like group A, group B, and all. Only one patient is a point. And as we know, we all say that I have given this medicine, and after 1 week patient was fine, or after 2 weeks the patient got significant relief. So we don't know whether the medicine is acting or the body is responding to the very minute amount of medicine what we have given, so that it is starting to, or it is this medicine is also acting like a placebo. We want to identify that. So in that case, we have to design in such a way. Once we can give a placebo, next comes a treatment. Then once you can give in 7 to 12 days, you can plan a treatment, then you can a placebo, then a treatment, a placebo, then you assess it. This is for our own assessment. It's like we all use it as a self-assessment of our treatments, whether this medicine is really working, or patient is impressing us, or I am overwhelmed with the drug reactions, or the. So all these points can be clarified, so that if in any platform we are able to answer most of the questions. So how do we begin with the research being a AYUSH doctors? So I would suggest that rather than everything, individual doctors must concentrate on case studies and case series. Because when we take up a case study or a case series, let me explain about a case study. Suppose I'm treating a disease, a rare disease, maybe like immunodeficiency diseases or compromised patients I'm taking, like tuberculosis or maybe immunothrombocytopenic purpura, like such cases, very rare case you are taking. We have to, whenever we take up such cases as a single case study, it's all our responsibility to identify what acharyas have told. So when we start explaining a lot of clinical trials and clinical studies, we are forgetting a basic principle that our science is built in such a way that there is a chance for individual drug classification. There is a chance for patient's age, patient body weight, and body, that is Prakriti is also there. Sara, all those Ashtasthana Parikshas, Dashavidha Parikshas are all included. And all the medicines what we are giving, whether they are on the basis of Shat Kriya Kala or Trivida Kriya Kala. So where we can document all those, we are unable to document them in case of modern clinical trial protocols. So we need to create our own protocols to identify these are the strong areas where we can work, and these are the our weak areas where others can work. So that will be possible only with a case study. If I'm treating a case of rheumatoid arthritis, a treating doctor, if he's from Kaya Chikitsa, he'll be knowing what is its nidana, what is its samprapti, what is its dosha, what is the dusha, then where comes its sthana samshaya, and which is the vekta stage, which is the bheda avastha, etc. So this algorithm can be made only by the Ayurveda doctors. And once we start giving the treatment protocol, it's not a single drug and single medicine, single disease, a theory in Ayurveda. It is always a protocol. So where if the nidana is so in X is a nidana of rheumatoid arthritis, so we have to, if we are considering it as a amavata, then we have to take up from all the nidanas, then we have to address them. In pathology, we have to break the pathology. In the medicines, what are the medicine is given? Why Simhanada Gugulu is considered among all the Gugulus? That answer we have to crack. Because Acharyas have not explained only Triphala Gugulu or Simhanada Gugulu for all the diseases, whatever they have told. Because each Gugulu is having a different combinations and they are having a different therapeutic value. So in our particular disease, which Gugulu I am selecting? Why I am selecting? In which stage I am selecting? Whether it is given in Pura Rupa Avastha, whether it is given in a Vekta Avastha. So this categorization we are failing to perform. So if you are looking very deeply into Charak Samhita or Susha Samhita, the whole protocol of a disease and its description and the management is based on it. Because I used to, as a student, when I was a student, I used to be always worried. Why? Or I was shocked to see how much medications, how much therapies, how much variations in the drug dosage are explained in a single chapter for a single disease. So there must be some rationality behind that. But we are all bypassing those rationalities and we are going towards a single drug and a single disease path, which has to be broken down. So to start that, we have to go with the case study. So this is my area of interest where I am working like a case studies. We'll not go because it's for all other postgraduate and PhD works, we'll go with all the clinical trials, which is required as a mandatory rule. But personally, we'll get a result, we can interpret and we can publish only when it is a case study, because we'll understand what exactly Ayurveda is about to say about that particular disease.
Your slides are not moving, Rajeshwari.
Yes, sir. I'm in the same slide, sir. Okay. Okay. So once that concept is clear, we have to go with the reporting a case, a new symptom, interesting finding, an important variation of the disease or normalcy, unexpected events or outcome automatically comes into our study. So this is actually a beauty of Ayurvedic descriptions, and it's step-by-step evaluations that as a clinician, as a researcher, we must do. So the case reports will give us a more strength compared to any other things. So in only if we are proving it with Ayurveda concepts, that is, everybody, wherever we go, the people will ask if we are speaking about some big diseases or unfamiliar diseases, they'll ask, what are the steps to begin a research as a clinical clinician? So these are the points we have to always remove. You can select any disease. What is that disease which can be found in contemporary medicines? What is the treatment algorithm that we can frame based on Charaka, Sushruta, Vagbhata? And what are the principles behind them in recent texts also, including Bhavaprakasha, Madhava Nidana and all? And we can make a treatment plan without violating the basic rules of the Chikitsa Siddhantas explain. And what's the result? And once we get the result, we can make it an outcome. So this is an outcome of one clinical case study. So if we are able to follow the same treatment protocols to all the patients, then it will become a clinical case series. But I would say that there will be slight variation from one patient to other patient. That's why one disease, one drug may not work here. Here also, because we have to look into Prakriti, Sara, Samhanina, all those points. So there will be mild variation in one patient. So all these observations are the scope, or it is the merits which we can claim in our publications or in our talks regarding our science. Because I don't like people just highlighting Guduchi is a anti-inflammatory drug. No, it is not an anti-inflammatory drug only, because it is told in Vata Rakta as a Agri Aushadha. Why it is told? We have to rule out. It is told as Jwarahara. That also we have to rule out. So Acharyas have not sticked on to one point. So all those dimensions are there. So one action of the drug and its benefit, and the name of the drug, its action, this kind of description may not be helpful for the actual development of Ayurveda. So coming to our field, that is Shalya Tantra, what is the requirement? It's about the innovations, because there are a lot of techniques which are present in the textbook, which may not be applicable right now. So it's a duty of each doctors or each clinicians to identify those areas which cannot be brought into practice. What was the reason behind explaining that? What was the idea behind explaining that? Because when we were in a BAMS, when we were taught that in Udara we have to do Rakta Mokshana from the cubital veins, that is in the arms, in the elbow. We have to remove Rakta Mokshana is done in the right hand limb, and in the splenomegaly we have to do in left limb. So this kind of description was available. Nobody gave us a answer. But after going to a lot of literatures, even in splenomegaly, because of the what do you call, the enlargement of the liver, there will be stress on the liver and also spleen, hepatic vessels, and also it leaves if the reason is just like what do you call, thrombocytosis, increase in the numbers of the platelets and also the corpuscular component of the blood. Even in the modern science, they are allowing us to go for a venesection. So that is a treatment is allowed. So in Udara Chikitsa, they have told means that it is not only the ascites that he was explaining, Acharya Sushruta, it may be something else also. So that thought we have to identify. Why when till now, even in the last year, I have seen patients going for venesection, and they are going for the blood transfusion or bloodletting in case of thrombocytosis. So this is there in the practice, but the interpretation is wrong. So those concepts must be given a new language of description, new language of understanding. So that will be one of the areas where we have to work. And the wound management, it's having a larger scope, because starting from the material, starting from the medications, we need to update. So all these, the modifications in the medicines also we have to make. Every time Acharyas have considered it as the kashayas can be used. Why you use only kashayas? Why you cannot make it into a most sustainable solution? We have to think of it. Take the references from Arka Prakasha. What Acharyas have told in Arka Prakasha, it is a non-preservative added solutions can be prepared for the complete wound management. So such thoughts, and what is the rationale behind using the green leaves in as a arka? Because we have seen all aromatic substances are used for the arka preparation, but in Arka Prakasha, if you see, it is not only the volatile oils are required. It's all the roots and also the stem and leaves of the plant is used for ARCA preparation. So what is the rationale? We don't have answers. So we are all teaching and we are taught like the ARCA preparation means it is to retain the volatile principles. So from that concise information we have to move. And then the surgical and para-surgical procedures should be modified for the safety, efficacy, sanitization. So many actions are there. So the people must work for this also in the closed circuits. So this is instead of just thinking about the community, thinking about the large scale research and clinical trials, wherever we are, we have to start with the with our own research. Whatever, it may not require a larger setup to start our own research, but it needs a new thought or new ideas also. Provided when we think all this, we must remember what are the mandatory knowledges we should have. So this is, we all will get these kinds of pharmacological studies, analytical studies, and how the interpretation of reports happens. What are the cell line studies? Is the preliminary clinical trial, whether our medicines are effective or not in the cells. So such studies are there, whether they can be brought into our trials so that our treatments or our medicines and their therapeutic value can be enhanced. What are the tools to develop Ayurveda treatments? Like if you want to prove how an external lepa is going to resolve a swelling, we need to find out how in Sushruta they have given a shloka. If in Pakvavastha, Amavastha, Pachamanavastha, what are the signs and symptoms will be there. So we have to tabulate that, and there are any instruments that also we have to look for. Vali Pradurbhavaha is there. When the swelling reduces, there will be a shrinking. That is, the skin becomes, the swelling reduces and the shrinking of the skin will happen. So there is a transdermal or dermal water loss is there. Is there an instrument to measure that? Yes, we have an instrument to measure that. So that kind of updations we must know. And one most important point is ADRs, adverse drug reactions, which is nobody is reporting, nobody is bothered. So such things should not happen. So we must, if we are applying some external applications, there will be some rashes. Yes, we must agree that it may be because of the medicines, or we have not used the base which is explained in the text. So those points we need to check. And also the last point is about publications. So when we take up both the sciences together, what is told in Ayurveda, what is there in the contemporary system of medicines, along with the what are the cell biology and other systems, how they can contribute to us. When all these knowledges are taken together, then it becomes a very good output to dispense or to exhibit in front of the scientists. So this will happen for a normal individual or a normal doctor who is sitting in a village or wherever. They can just work with the in his own clinic. So that is very important. That is very easy also. So we do not need a bigger research setups. So it needs a new thought. So what is the benefit of all these? When we speak, yes, I'm treating this medicine. I'm treating this disease with this medicine. We have an evidence. How? Obviously, the people will ask how this is going to affect. So all this how can be answered when we can list out, or when we can step by step, when we categorize with the Ayurveda principles and we match and we use the modern or the available techniques to prove that. And never forget to report or to identify the drug reactions also, and do conclude it with the there is no rule is for like single drug and single disease for Ayurveda. So these are the points which we have to remember when we are performing a research in case of in Ayush. So that's why both the science must go parallelly. So that's what I would say. With this, I'm concluding my session, and thank you for all the organizers for the opportunity. Thank you.
Thank you, madam. Thank you. Very brief and to the point Ayurvedic discussion. Excellent. Now I would like to call Dr. Arun Raj from Hassan. Sorry, Dr. Arun Raj. The link was sent delayed. Welcome. Welcome, Dr. Arun Raj. Dr. Arun Raj. Am I audible to you? Arun Raj GR. Excellent, Rajeshwari ji. You have completed excellent. Dr. Arun Raj.
Thank you, sir. Thank you.
Excellent, madam. Excellent. Dr. Payodhi Dhar, am I audible to you?
Yes, you are audible.
Welcome, welcome, doctor. Welcome. Yes, kindly continue, Dr. Payodhi ji.
Yeah. So, good evening, everyone. It has been an honor to be invited by our very beloved, my senior Dada, leader in the field, and a man of so much enthusiasm, Dr. Pawan Sharma ji, by here. Unfortunately, I am out of the city now, so I could not connect earlier due to the network issues and all. But today's topic is very, very important for all of us. Why? Because when we are talking about the clinical trial and good clinical practice, this does not mean a particular habit only which is confined to the people of our community of doctors or something like that. It involves a hell lot of industry. It involves the people who are actually studying pharmacy, the production guys, the guys who are involved in the publicity of it, and so on and so forth.
The voice is continued, Dr. Payodhi.
Your voice is not coming, Dr. Payodhi.
Am I audible?
Now audible.
Am I audible?
Yes, now audible. Kindly continue.
Sure. So what I was, I don't know from why you could not hear. Actually, what I was trying to say that this topic is very, very important for not only the doctor community, but also the healthcare people at large, the pharmacists, the pharmaceutical guys who are studying it, the production house, sponsors, researchers, biomedical researchers, biostatistics guys, and everyone. Why? Because it's an encapsulated approach to giving best possible available and affordable healthcare services for today and also the future. In the world, India is now one of the gurus of the healthcare industry and the pharma industry. We are supplying medicines and all. And also, it must be noted that we have a good amount of patient pool, which is being explored ethically, who should have been explored ethically, but at times not that much ethically by many pharmaceutical industries, many a times, which we need to be careful. Here, starting from BABE study centers to the CROs, SMOs, and everything, this can be created. When I am talking about these issues, I know the previous speakers and next ones will discuss about the technical integrities of it. What I want to emphasize is the scope of career for the new coming people, the scope of industry development, and creating employment as well as influencing on our economy by this modality. So good clinical practice is something which will not only ensure a safe and efficacious drug at an affordable price, but also it will ensure the quality of research, the amount of medical data which are required for better and better development of affordable, less costly, available for all drugs. In this regard, we must understand that there are three parties which are involved in it. Number one, sponsor. Number two, the investigator. And number three, the auditor. In the sense of auditor, I mean the ethics committee, who need to know the answer, the question, why the research is done, next, how the research is being done, and that means the protocol of research, and last but not the least, to check on every single point of the process of research so that we can come out with a good quality of research. In India, there are very few committees which are available. There are some institutions who have the institutional ethics committees, whereas there are some bodies who are coming up with independent ethics committees. Now, in order to create more and more research available, for which the fund is there, we need to have some proper centers. We need to create many more independent ethics committees available. And also we should emphasize on good research. Try and understand when research is being done, it's not merely answering a question or merely finding out whether this drug works or not. It actually deals with a quality of medical data which will have an impact today and also tomorrow, will enthuse or change the track of many decisions which might be clinically very much important, might be fatal, might be life-saving. Let me give you an example. Empagliflozin is a drug which is used, which is a sort of miracle drug in diabetes right now. Few years back, some people used to say that this is a drug which acts through kidney. It leads to higher amount of urinary tract infection. It is bad for kidney. You should not be giving it to anybody, something like that. That was the notion which used to be there. The other competitor companies used to sell that, listen, you should not give this drug to any renal compromised patients. But then the renal study of it, something. Am I audible?
Yes, yes. Please
What did
continue.
they find out? They did find out that this drug not only helps in decreasing the chance of progression of renopathy, but also it might be somewhat renoprotective. So the data which we got changed the entire paradigm of what we have been thinking till date. Next, when the changes came in some other drug like bioglitazone, it is a very good drug which was being used for treating diabetes. The market started telling us suddenly that it leads to bladder cancer. And one fine morning, everybody stopped writing the drug is gone, so many bad things being told about it. But suddenly, one thing was found out that
Your voice is breaking in intervals. Please take care, Dr. Payodhi. Your voice is not available. Hari Om.
now available.
Yes, kindly continue and conclude.
Okay.
Because in intervals, voice is breaking.
Okay. There might be some. So, could you hear the thing I told about this bioglitazone and all, the bladder cancer theory?
Yes.
Okay. Okay. But it's very interesting to find out that this theory, that this bladder cancer is a myth and people are selling it just to, carve this good drug, was found out by some independent ethics committees. And as they did find out, people tried to rule them out. But in the long run, it was found out that there is no such thing like increasing bladder cancer, at least not very much prominently, not significant. Now the point of significance, what is significant in research and what is not? There biostatistics came in, the p-values came in, type of studies came in. Then we started understanding that when we are reading a data, any medical data or information, we need to understand from which category of research is this coming out. Suppose a research which is being done in a non-blinded study will be always inferior to a research which is done by blinding of study. Then comes double blinding, triple blinding. What are they? But the participant and the researchers are both of them are unaware of which drug is being given to which group. Then comes randomized controlled trial, RCT. Then comes meta-analysis, let's say. It's just I'm telling schematically. So meta-analysis gives you a data which is most authentic. Then comes RCT. Then this blinding, double blinding, single blinding, no blinding, like that. So when we are appreciating a data or any information, in our understanding, it is important to understand which category of data is, which type of research has been done, how that research has been conducted. Suppose this research is being conducted by some person who has some interest, vested interest in it. So conflict of interest in research is another very important thing, which is absolutely related to good clinical practice. Now, good clinical practice does not only take care of these few issues. We need to understand that. The clinical research scenario and good clinical practice does also take care of how the authorities will act to you when you are doing a research. It's there in 2019 new clinical trial rules. In today's world, India is trying to come up with self-reliant production houses. In manufacturing and all subsidies are being given by the government of India. So what is they're trying to do in addition to the normal things, that if you submit the research proposal, within 90 days, you will get reply from them. If you don't get, it is implied that your approval has been given. Then on, when it came forward, they have set up some lines, both from WHO and from others, how should a research organization behave? What are the functions of CRO? How should they train their staffs? How 12 to 20 codes of GCP should be maintained? All these things are here. But then comes another important point, that is adverse drug reaction, unwanted death, and so many things. Now, about this, pharmacovigilance have taken up a very, very important place. For the people who are studying pharmacy now, they might have options of only the industry or teaching few years back. But now they have scopes in biomedical research. They have also scope in pharmacovigilance, which can pay them as good as industry. Now, this pharmacovigilance and reporting system, PVPI, has given us many things about many drugs. Also, we have come to know that how one person should approach when we are dealing with the adverse reactions, how that should be categorized, which is desirable, which is non-desirable. We have understood that side effects, adverse effects, and adverse drug reactions, these three are three different terms. May I repeat? Side effects, adverse effects, and adverse drug reactions. These three things are three different categories of terms. Now, these all come under the topic of good clinical practice and clinical trial rule. So we must understand that clinical trial is a huge thing now, which has a huge amount of fund flowing from outside. We have already the database, we have Indian data, we have people, we have patients, we have skilled doctors, we have researchers, all the things which are required, money, manpower, and the material are there, but we need to coordinate it. I must congratulate Dr. Pawan Sharma for arranging these things for long. He has been doing it by his own independent ethics committee. I have been associated with that as committee too. How he takes care of things, the way he deals with the biostatistics, the protocol writing from the point of protocol writing and all, that's excellent. He has already, to my knowledge, has appointed a girl who has passed pharmacy, and he has been trying it singularly. I feel we must all join hands with him and make this a practice. This model of Medpharma CRO should be replicable and scalable throughout pan India. Many things can be said in clinical research, but with this, I'll conclude here, and my best wishes to Dr. Sharma. Thank you.
Thank you, Dr. Payod. You are excellent as always. Really excellent. Please do join our team of AYUSH Samriddhi regularly. We wish you all the best for your future. Thank you. Thank you very much. Now I would like to request Dr. Arun Raj from Hassan. He is also from Kumar Bratya. Welcome. Welcome, Dr. Arun Raj. Welcome. Please unmute you. Yes, welcome.
Good evening, sir. Kindly make me co-host so that I can share.
Yes, please continue. I am thankful to Dr. Swaz Sethi, Director of KLE Belgavi, for introducing Dr. Arun Raj. Welcome. Welcome, Dr.
Welcome.
Your screen is visible. You can go for slides. Yes, screen is visible. You can go for the slideshow to maximize it.
Yeah. Is this clear? Am I clear, sir?
Yes, yes. Yes, please continue.
Yes, yes.
Okay. Right. Much of the practical aspects of regarding clinical research and various types of study designs. So I will invite all the dear delegates, and I will be the next 10 to 15 minutes. I'll be talking on the various study designs that we can use in clinical research. To be precise, I will take you to a tour towards the epidemiological study designs, right? So as such, at the end of this module, we will be able to list the different study designs that can be used in a particular epidemiological program scenario. Also, we will be able to select an appropriate design for a particular type of research. Most of the time in Ayurvedic research, so we will be confused like which type of study design and we have to go for, right? So at the end of this lecture, I hope we'll be able to, right? Now there are, in general, we have two types of methods approach. One is quantitative and qualitative. Throughout the Ayurvedic researchers, most we will be using the quantitative methods, like which, the earlier resource person was highlighting. We are focused on the p-values, where we focus on the quantity, that the numbers where you measure, and which you make use of the statistical softwares for our evaluations. On the other hand, that's our quantitative methods. Second, we have the qualitative methods, where, for example, we have the ethnographic studies, which runs into qualitative types of methods, and one majority will do the qualitative methods, right? Now, the study designs in a nutshell, in the epidemiological studies, we have two considerations. One is observational, second is interventional. So I will be giving a gist of the type of study designs, so we can have a further discussion later in the coming series, right? In the observational, we will be considering both descriptive and analytical. So we have in the descriptive case reports for a single case designs, ecological type of trials and cross-sectional. Now in analytical, we have cross-sectional case control cohort, and interventional, yes, some previous resource person mentioning regarding RCT. Also, we have the field trials and the community trials. While framing a research question, there are three things that we have to keep in mind while framing a research question, right? So first is that you keep an eye on the distribution of the problem that you want to investigate, right? So you will focus it based on the time, place, person, and you will use mostly the descriptive study designs. For example, now we'll assess the prevalence of severe. Am I in the 6th slide?
No, you are on the first slide.
Okay. Sir, is this okay now?
Okay.
Okay. Right. Right. Whether I am on the 7th slide now, sir? Pawan, sir?
Yes, please continue. Yes.
Yeah, thank you so much. Second, we have the determinants. So,
But
right,
go for slideshow to maximize the slide.
Yeah.
It is on PowerPoint mode. Please go for slideshow.
Yeah, is this now? Is it moving? 6, 7, 6, 7?
It is moving, but it is on PowerPoint mode. Please go for slideshow. Presentation mode. Yes.
Yeah. Right. Okay. So second,
Excellent.
based on,
Excellent.
Thank you, sir. Second is based on, one is the what is the distribution, and second based on the determinant. So distribution, right? First one was distribution, right? I was speaking on distribution. So for example, right, we may make a research question like what is the prevalence of malnutrition in a particular community, right? So, or which age group of women have higher burden of unsafe abortions, right? So which state in India has the maximum infant mortality rates? That is one type of framing a research question. Second is, I think I am on 7th slide, sir, right? So determinants. Now, determinants is usually studied by using analytical designs where the factors, what factors are causing the particular disease or the problem. So for example, why is the NMR stagnant for the past 10 years, right? So like this you can frame. So why is the decline in MMR faster in Tamil Nadu as compared to AYUSH? So these are some examples of research questions that we can form. Third is mostly we will use it in the effectiveness of a particular intervention to understand Ayurvedic study designs, which treatment is better, which type of intervention is better. Right. So what works better for childhood pneumonia, cotrimazole or amoxicillin? So that's an example. Is folic acid supplementation effective in preventing open NTDs? So these are the various considerations of framing a research question. Now, the first type of study in this one is the observational studies, where we allows the nature to take its course, and the investigator will be measure, but does not intervene. So it once again have two aspects. One is descriptive and analytical. So where in analytical we go for analyzing the relationships, right? So the first of its kind is the case reports. So most of you might have done, right? So one, you report a single case, which the experience of a single patient. So we document the unusual medical occurrences which happens, which gives a first clue of a particular identification of a new disease or an adverse effect, right? Right. So that is regarding a case reports. So I'll be rushing through this particular, all the study designs. Second is a case series, when you have a collection of individual case reports, right? So which occurs within a short period of time, right? So similar, then these are very useful in formulating a hypothesis, this case reports, and after that the case series. So both these falls under observational type. And there are some advantages and disadvantages for case reports and case series, where the advantages of case reports and case series are, it is anecdotal, always anecdotal in nature, and it's used to create a hypothesis, right, for a future planned control studies. And the disadvantages is, and usually it will not be representative of the population. That's why we given a particular Ayurvedic treatment to a patient, it's shown relief, but in the next page it will not be. So that is one disadvantage, and always in research the comparison is having weightage. So it's the comparison group will not be. So that is another disadvantage, right? Now, very less we use this ecological studies in Ayurvedic context. For example, a study I want like an example, right? Where you find out the association between institutional deliveries and maternal deaths. Right, for example, so such type of studies are also can be taken up in Ayurvedic research setup as part of ecological studies. And the advantage is it is like in case reports and case series, it is trying to generate a hypothesis for future. But the disadvantage is there will not be a individual level information, right? And you can't determine the causation in this particular ecological type of study designs. Now, the basic epidemiological design is the three, like we will be using the case control, cohort, and cross-section. I devote some time to this slide particularly, where in cross-sectional we have the disease and the exposure at the same time you will be dealing with. But in cohort, present you will be having exposure, and you will go to the future disease causation. We'll see when the disease will be occurred, right? The exposure cause for how much percentage of the exposure can cause for disease. And in case control, in the current scenario, we'll be having a disease at hand and we'll go to cohort. Previous, right? You go see how from which type of exposure the disease might have occurred in what quantity, right? Like as the cohort is, you from the exposure you go towards the outcome. In case control, from the outcome to the exposure. While at the cross-sectional type of study, we will deal with both the exposure and the outcome in the same scenario, same platform. Right. So rather these cross-section studies are referred as prevalence studies, right? So we will be invariably using the administering this type of studies in Ayurvedic researchers, where you measure the exposure and effect at the same time, at the snapshot, right? And which is these are very easy and inexpensive. And the tools that we need is a structured type of questionnaires, right? And then with a good sample size and a good response rate, and which will be helpful in assessing the needs of the healthcare needs of a population, right? And these are very indicative of earlier trends. And it will provide, in this case, cross-section studies will provide information on the prevalence of a disease and health outcome. Most of the time, our students have the issues with, sir, we didn't got the prevalence of the particular disease, so we will go take the prevalence from Brazil, right? So better that we will be conducting a cross-section studies which indicate what the prevalence of the particular population, and then go for the interventions of particular interventions, right? And it also allows to create a hypothesis, and the disadvantage is this type of studies which gather the prevalence but not the incidence. That is a disadvantage, right? Yes, that is the one regarding cross-sectional. And then right, in case control, yes, I told we have the disease at hand. We will go back and see how the it investigates the association between the factors which led to the development of the disease. So how which type of exposure has caused the particular disease. So for example, of a case control study, right, to find the association between diarrhea among children under 2 years with breastfeeding. So we'll be at the end, what we'll be getting, we'll be analyzing the exposure rate among cases and controls to breastfeeding. How much percentage of the children who has breastfeeding has got diarrhea, how the breastfeeding has contributed to diarrhea, using a questionnaire which is inclusive of breastfeeding history, duration, all those things, right? Then now here comes our first calculation, where you use the odds ratio. Like we have the you have two samples. We have the exposed population and the unexposed population. In that you designate A, B, C, D, right? You have the case and the controls in the exposed population and unexposed population. Using the odds ratio, we'll calculate, right? So if in the odds ratio, if it is odds ratio is 1, there will be an association with the disease and the exposure, and the odds ratio is more than 1, there will be a higher risk of exposure. And if odds ratio is less than 1, we conclude it as there is a lower risk of exposure in the diseased condition, right? So this is how we calculate using a case control study. And this is quite quick and inexpensive. Due to time constraint, I am rushing through all the points. The strength is it's very easily quick and inexpensive, and it is it's an optimum for evaluation of relatively rare diseases. So that is the positive points of this case control study, right? So on the contrary, the cohort study, it's a longitudinal one which is forward looking, right? You have an exposure at hand, and you will further you make a longitudinal observation study to forward looking to see how this particular exposure will cause for the disease. Now, so where in you have a group of people which having common characters in this this one, and contrast to this case control study. Here in this case, we will be selecting cases with a particular disease, and they try to ascertain their exposure status to specific risk factors for developing the disease. And now you will be having following these particular steps while conducting a cohort study. Like in a case control, we will be having an objective, and then you select the cases as well as controls, and you obtain the data, follow up and analyze. This is in a nutshell. Right. Now here we have a calculation of risk ratio or relative risk. Now, similarly, like in case control study, if the odds ratio, like I told you, in odds ratio case control 1, value is 1, it says no association. Like in relative risk or risk ratio, if the value is 1, there is no association. And if relative risk or relative risk ratio is more than 1, there is an increased disease risk among the individuals. And then if it is less than 1, there is decrease. We'll conclude it. That's how it's calculating. So I have put an example over here, right? So it is a positive is it proceeds from cause to effect. That's a very positive thing, and it can yield information about more than one disease outcome because you are following it for a. But that is one negative limitation also, because you will be having a long follow-up period for this particular type of, and it's always expensive in that context. But the quantum of data that they generate is limitless. And in the other hand of the epidemiological studies, to conclude it, we have the RCTs, field trials and community trials, right, in interventional. And so where you actively intervene in this experimental type of studies, right, you actually have an active attempt to change the sequence of a disease using an exposure, using an intervention, right, that you will regularly use in Ayurvedic type of studies. I think all are aware of it. And then we have RCTs are considered to be the gold stone. I think the previous people have previous resources have talked extensively regarding the types of RCTs and everything. So RCTs are considered to be the gold standard of all types of in the clinical research. And then we have the community trials, field trials, where in nutshell, you assess the patient during enrollment for the eligibility, especially in RCTs, where you assess for the eligibility, and from that you exclude the patients who does not meet the inclusion criteria or who decline to participate or any other reasons, and then you randomize it using any techniques like, for example, like computer generated tables, etc. And then we can allocate into 2 groups or 3 groups or any number of groups, right? And then then we will be following the patient, and then you analyze it. So this is in a nutshell how you can frame a type of RCT or field trials. And I think I have minimized myself to the key points of like epidemiological studies, starting from case reports, case series, ecological, cross-sectional, case control, cohort, and RCTs. Now, what should be our consideration on the last slide? is choosing the study design is so important. Like to prepare a home, choosing the a suitable plan based on your budget. So like this, we should be considering whether this study design address my question that you have formulated based on determinants, etc. And is this study design ethical? Right, that's so important, especially those who work with pedia. I am from a pedia, that's why. So what resources do I have? Time, money, personal. It will be varying based on it's a project or funded. If it is non-funded, self-funded, or it's a PG research work, you'll be getting hardly 1 year to time. In PhD work, you will be getting limitless personal, all those things. So what resources do I have? And is there a cheaper and quicker way of answering the same question? So based on all these things, we should to conclude, we will be considering a study design, because study design is the basis. Once that's why I selected this topic, because once the study design is shaky, nothing will come proper, right? So I think I like to wind up within this 15 minutes. I thank Dr. Pavan Sharma sir for this opportunity and your association, and thank you so much and good night.
Thank you. Thank you very much, Dr. Arun. Thank you very much.
Thank you, sir.
Very briefly, you have communicated all things about study designs. I am really thankful to all the speakers. Now I would like to request Professor V.K. Agnihotri sir to moderate the open session. Welcome, sir.
Thank you, Dr. Pawan Sharma ji. Namaskar. As we have all the 4 distinguished lectures completed, and after Professor Renu Rathi, we have got 3 more speakers who presented our speech. The one is Dr. Rajeshri, and 2 others are Dr. Payodhi and again Dr. Arun Rajji. Very naturally, it's a pleasure to say that excellent presentation was by Dr. Rajeshri ji. Very simple and very confidently, and really she removed my all the illusions and doubts which I was talking about the any Ayurvedic setup for the research in AYUSH section. She also raised a very important point when she was presenting, when she was studying. I very much liked that point. I noted down, I underlined that it was she, or rather it was her mind, that why this dose system is there in our text, and if we are treating the patient at the level of say Pur Rupavastha or Prak Pur Rupavastha or Rupavastha and full-fledged say disease. She talked and her answer is also there in her question itself. Why the dosing system is 2 to 3 tablets 2 to 3 times a day. That itself reveals the situation. It is up to you whether it is 125 mg or 250 mg as per the need. If you proceed with as per the intensity of the symptoms on the basis of the diagnosis, if you proceed with for 1 week, see the percentage of response. If you are feeling that it is not up to the dose given to the patient, you can revise the dose. So that was a range 2 to 3 tablets or 1 to 2 tablets 3 to 4 times daily. This is here our texts are very clear. So her question and answer in the same way, but very, very intelligently she presented in that way. And Dr. Payodhi, as usual, Dr. Payodhi and Dr. Arun, what happened? They are all attached with the completely research work. So total research methodology, which is chalked out for MD scholars and PhD scholars, are equally narrated by them. And very naturally, since we have to be in the field, we have to go for the same parameters and protocols to chalk out the disease, because medicines are there, text is there, rest the students, research scholars are supposed to meet out. So with these remarks for all the 4 distinguished guest speakers, now I, as per our instructions, as per instruction to me by Professor Pawan Sharma ji, now I start the group discussion. And meanwhile, I am remembering that once Dr. Nishant Shukla ji raised his hand, so he might be having some very important point during the presentation of first or second. So I very honorably welcome Professor Nishant Shukla ji. Please come. We invite you to come with your suggestions, questions, doubt, or anything add-on you want. Please. Namaskar. Shukla is here. If Professor Shukla take times, I would like to have a few new faces are there. Either they are with our say the good students or research scholars or the doctors themselves of our distinguished guest speakers. But I would like to start with Professor P. Sudhakar Reddy ji. He is a very senior person, so he will definitely give his experiences or the checking out the works where the research work is not oriented on medicines or based on some of the formulations. Yes, please. We invite you, Professor Reddy sir. Professor Sudhakar Reddy ji. Are you?
Namaskar.
Namaskar. Namaskar. Yeah, namaskar ji.
So good evening to all. Dr. Sharma sir, Dr. Agnihotra sir, and all other speakers. Today's webinar was a very excellent webinar, sir, and need of our, especially for Ayurvedic faculty members. Nowadays, in all institutions and all over medical colleges also, Ayurvedic colleges also, the research is a part and parcel. So everyone speaks about the research. Now there it comes more than animal experimentation. We are behind of the clinical research. And if you see that all the samhitas, total Ayurveda is clinical research only. Because without research, they could have not told that this is best treatment, this is again best procedure for this disease, etc. So if you see in a deep manner, the total Ayurveda, whatever Chikitsa mentioned by Acharyas, that was only clinical research was there in previously. But if you say this one, the modern or the scientist, present scientist may not believe.
Yes. Sir, आपकी आवाज रुक गई. Professor Reddy, your voice is discontinued.
After 5 minutes, sir.
Yes, yes, please.
So I'll speak after 5 minutes, sir. Just sorry for disturbance.
Okay. You will join again. Meanwhile, Professor Shukla is with us. So I welcome Professor Nishan Shukla, sir. Professor Shukla ji, Nishan Shukla ji. You're also gone. Dr. Harish Sharma ji, are you here? It's 9:45. How they have left so early?
If anybody would like to add anything, welcome.
Yeah.
In clinical researches, welcome. Reddy ji is rejoined. Nishant Shukla ji is rejoined. Am I audible to Nishant ji? Dr. Nishant Shukla ji. Welcome.
This was really very nice presentation. I was having some issue about the network, and that's why I was not able to hear some of the parts of the few presentations. This is actually the needed part. We need to differentiate between the clinical study, the clinical trials, objectives of the clinical trials, what happens in our work of MD and PhD, doctoral research. We take too many objectives or a very broad objective, and that's why that is creating the problem for getting the proper outcome of the research. That is a big lacuna. So we need to focus very precisely on the research design and this is my opinion, that we need to focus more on research design. In the present day context, the world is looking for the importance of well-being. And this is narrated in Ayurveda in the context of Dehabala, Chittabala, and Indriyabala at the time of doing the management of the or the treatment. One should observe about the efficacy of the medicine on the clinical parameters, the clinical evidences and the markers, the signs, symptoms and the markers. And along with that one, the researcher ought to look into these important functions of the body. So this for the well-being and the research. This is the highlighting feature I would like to add to these presentations. I am thankful to Agnihotri ji. I am thankful to Sharma ji for giving me chance to speak on this issue.
Thank you, sir. Thank you so much. As per Professor Pawan Sharma's appeal, it will be good if voluntarily those who want to add on something or suggestion, questions, you please come so that our time will be saved. Dr. Mallikarjun Rao, sir, would you like something? Add?
Sir, I have just now joined. I was unaware
Yeah,
of things.
you have not gone through. You have not gone
Yes,
through
sir.
the lecture. Okay.
Just
No
now
problem.
I joined, sir.
Okay, okay. It will
Yes,
be
sir.
a pleasure if I just invite our guest speakers as they are still with us. So any feedback or anything for 1 minute they want to add on something. Meanwhile, after the hearing all others discussions and presentations, Madam Rathi and Madam Raishri. Yes, please. Would you like to something add?
Sir, yeah, it's difficult to get rare cases as my next presenter was insisted on only case report or case series. But I am on that whenever we do particular research, because in practice it's very difficult and it's focused on case report or case series. It's easy in practice, but in academic institutions we have to go and we have to conduct PG and PhD scholars to go with RCTs only. So in both way, it's again, it's a good part that someone has some presenter has focused on another point, someone has focused on another points. In that way, all of the parts are covered. But I think that it's very difficult in Ayurveda perspective to go ahead with clinical research, because it's a individualized science, and many times it's difficult to fix on criteria, because it's yukti or desh, kal, vay, bal, rog bal, rogi bal, prakriti. In all aspects, it is getting verified always. So many times we need to it's difficult to stick on one parameter or one intervention or one all set of conditions. So practically we come across with so many difficulties. But as we consider about modern criteria and modern study designs, and those are little bit easier as compared to Ayurveda, because we generally we don't stick to our pramanas, because they are not evident so much as compared to other modern faculties mindset, because they are totally unaware about our pramanas, our aptopadesh, our yukti or anuman. So they are in the view of that only their mindset, that we require so and so evidence, then only we consider it as a good one. So in Ayurveda perspective, clinical research is a little bit difficult, especially because it's an individualized science in my view. Okay, thank you so much.
Thank you, ma'am. Thank you so much. For one thing, I am fully confident that the way you presented today, and I have heard you earlier also, you might be teaching to students very nicely. very confidently, very nicely. This I found on your presentation. So easygoing. It was not feeling that you are reading anything or you are taking any help of that free flow extempore. So thank you so much. I want to say thank you so much. I want to say something. If Dr. Arunji and Dr. Paladiji would like to say anything on the point raised by Dr. Rajeshri, because he was listening. Dr. Arunji or Dr. Fayodiji, would you like to say something that on the basis of the Prakriti decision and other Rasguni Vipak or treating a patient, the patient who is being treated with your treatment in clinical could be put under the say research module, and would he like to cooperate you for the period you are going to have a trial, say 3 weeks or 1.5 months or 3 months, like that? If at all his disease continues, if he gets okay, it's wonderful. So anything which is raised by Dr. Raishi Dr. Arunji, if you would like to say something, or Dr. Payodiji, if you can say something on this issue. I think Dr. Sudhakar Reddy ji now came with a better presentation, visualizing us very young and very fresh to say something. You are enjoying summer, and here we are suffering from severe cold. It is 6. And you are in Mysore.
Thank you.
मजे ले रहे हैं आप मजे ले रहे हैं मैसूर के.
Nice, sir. Sir, sir, I would like to first.
You are continuing something. You are saying something. Please continue. Would you like to add on something? Am I audible? Your voice is not available. Audible. Dr. Rathiji, yes. If you like to say something, because I would not like to carry on today. It's a shivering over this webinar. I want to finish before 10, and almost the total arc and stack of a webinar is being received by us through the mouth of our very intelligent guest speakers. Yes. Unmute kijiye. Please unmute.
Sir.
Yes.
Some network issues are there, sir.
Exactly.
Dr. Renuji was my colleague's wife, Dr. Bharat Kumar Rathi.
Yes.
He was my classmate.
That's why she is using Renu Bharat Rathi. I was just thinking, though. Yes. I got it. Because in banner it was simply Dr. Now she is using Dr. Bharat Rathi's phone, I think, because now the name is here, Dr. Bharat Rathi.
Bharat Kumar Rathi, my classmate in PG.
That's why I got that number of persons from Mysore might be joining on her presentation. Yes, please. You are saying something.
No, sir, I was regarding one question like Rajeshwari was speaking and other speakers just spoke regarding the RCTs, randomized clinical trials. Ayurveda, especially speaking, it's difficult to conduct RCTs, even though RCTs are the gold standard according to modern research methodology. If we conduct a research in an RCT and double blind, it is gold standard. It's scientifically accepted all over the world. Still, by using Ayurvedic methodology, it is all cases difficult RCT. That's why we can go for case studies or case reports. Even if you go for black box design, which was discussed by Dr. Rajeshwari. The black box design, recently, WHO have framed this black box design for traditional systems of medicine like Ayurveda. In Ayurveda, we are using for a disease 2 procedures, one procedure and 1 drug. So there, the problem will come in modern medicine. So they are using only 1 procedure or 1 drug or 2 drugs of same quality. But we are using a drug, a procedure combinedly, like Bhastikarma we are doing, and also efficacy of some other drug. So that will not be possible in modern RCTs. Exactly. Black box design, which is developed by WHO, it suits for Ayurvedic methodology. That we can say. And basically, the researcher, when they start clinical study, a case study, case reports, then we can go on up. But evidence-based medicine needs the RCT and meta-analysis. So that's why they are saying that Ayurveda should be evidence-based, means we should conduct the RCT. And low level of evidence comes from the case reports, case series, cohort studies, case control studies. So in Ayurveda, we can conduct from the down level only. The case control studies again little difficult, and cohort studies are again, because in cohort studies, retrospective study, okay, we can gather information from the patients which are already taken treatment, we can compare it. But prospective longitudinal studies needs longer duration. That will be possible only institutional level, that is not from the PhD or PG scholars. That institutional people, they should take up their studies for long studies, cohort studies. That can be censored. And otherwise, all I think they have covered everything. Even Arunraj also covered all the research methodology. If anybody by chance, if in the audience, if PG scholars are there or other people are there, they will be getting good knowledge. I hope somebody may be there in the our webinar today. They will be getting good knowledge regarding clinical research from down to earth, sir. All they spoke nicely. Thank you very much for giving this opportunity.
Thank you,
Another
Professor
point,
Reddy. I think
I would like to,
yes,
yeah,
please,
I would
yes,
like
please.
to one point that pragmatic trial has been left. So pragmatic trial is a very good tool in public domain. Whenever we do practice and it's individualized science, we can take 2 to 3 interventions and we can see that for one particular disease, how that 2 to 3 interventions are going to affect with multiple patients. So the size is big little bit, but we can see that which intervention is working very effectively in that particular patient. Or we can categorize patient that this area, this drug is more evident pool or working more nicely, and we can categorize the intervention as per race or religion or like that, or maybe Prakriti specially or Agni. In that way, a pragmatic trial is very good and especially Ayurveda perspective. Thank you.
Thank you. Thank to both of the guest speakers and our discussion panel persons like Professor Dr. Reddy, Dr. Renu Rathi. You see, I would like to go with the stand of Professor Reddy. As he pointed out, in fact, what we are doing for last 30 plus years, we are giving medicine to our patients. Of course, it is undocumented, but we are getting the influences out of the response they are getting out of our prescription. It is going on with all the clinicians, with all the persons treating the patient, even in practice, in private practice or in government sector. We know our next visit of the patient itself is a real card holder for us. That is दवाई ने काम की, इतना मुझे फायदा हुआ, मेरी उल्टियां बंद हो गई या मेरे पेट में दर्द उसके बाद नहीं हुआ, बट मुझे अभी दवाई चाहिए. क्यों? क्योंकि अभी हम 100% क्लियर नहीं है. So this was we are doing, we all physicians doing for last decades of and plus 35 years is with me. So I will go with the stand of Dr. Reddy. He really pointed out that in Ayurvedic concept, we have to have a case study, drug trial for his benefit, for his well-being. Now comes the scope of research work with the MD scholars. They are also visiting their clinical duty right from the 3rd year, 4th year, internship, then starts the 1st year MD, 2nd year, 3rd year. And meanwhile, they are also well versed with this procedure, and they have a patient module, and they check out the program. So Professor Reddy's concept is very practical and clear. Thank you so much, both the guys. Thank you.
Thank you, sir. Thank you very much.
Thank you all.
With these remarks, if any person would like to say anything, or Pawan Sharma ji, is over to Pawan Sharma ji back. Yes, sir.
Thank you, sir. Thank you very much. Today, really, seminar was excellent, sir. And now I hope no
Yes, you
question,
want to
no
say something?
suggestion is from audience. All the speakers have clearly communicated their views regarding clinical trial. Definitely, the subject is vast and it cannot be summarized within 20 minutes of the lecture. So I will be traveling all the speakers again and again on specific points of clinical research. Thank you, thank you,
And
thank you very
our
much.
guest speakers are very cheerful. So they are much pleased with today's webinar, what I infer out of this whole clinical trial. Or rather, webinar trial. Out of the webinar trial, we got a good assets on today's webinar. Thank you so much for giving your
Please
knowledgeful lecture.
please communicate me who is ready to talk on BF.7. I would like to create one video tomorrow. Please guide me out of audience and speaker who is ready for BF.7 and remedies from Ayurveda. Possible remedies for Ayurveda. Nobody has tested.
Ready or ready? We have got ready. We have to search for ready.
No, sir. That is a separate issue, sir. Who want to talk, please communicate on my WhatsApp. I will arrange one video tomorrow on details of BF7 and the possible treatment through our Ayurveda or Ayush. Thank you very much. Otherwise, BF.7, excluding BF.7, we are meeting day after tomorrow on Farmers Wellness. Thank you. Kisan Divas is there on 23rd. So thank you very much for joining. Please do join as today. Thank you. Good night. Good night.
Good night. Good night.
Good
Sir,
night.
can you repeat that, sir? Tomorrow's video. It's word.
BF.7 and possible treatment, line of treatment in our Ayurveda.
Okay, sir. Right, sir. Thank you, sir. Thank you very much.
I will collect all details on BF7 and somebody should guide me on Ayurvedic. So 2 speakers, one me and one Ayurvedic treatment. So please guide me on WhatsApp. We will arrange tomorrow.
Thank you, sir.
Thank you, because it is